Hello.
Good morning.
Good morning.
Good morning.
We'll be just starting after a short while. We are just waiting for our Honorable Vice Chancellor.
Yes, Thapa, I am here.
Okay, sir. So then let's... Good morning, everybody. A very warm welcome from Dinhata College and Cooch, Panchanan, Barma University to all. At the very outset, we have the privilege to briefly mention that due to the effect of COVID-19 global pandemic, there has been a sea change in all aspects of society, and this education is one of the worst victims. I recall the words of our Vice Chancellor, Honorable Vice Chancellor, Dr. Kumar Mukhopadhyay. During our last webinar, he has said that this pandemic effect is very high in our education sector, especially the areas where there is no technology and to some extent where there is a low technology like ours. And eventually after that we have thought that how best and how fast we could cope with the situation, and found that these online activities could streamline our teaching learning processes till we attain our conventional mode of face-to-face learning system. With these motives, we have ventured to organize this international colloquium on science and society, institutional approaches during pandemic by Dinhata College in collaboration with the Department of Botany, Cooch, Panchanan, Barma University. The august presence of our Vice Chancellor as the chief patron and respected Vaishna sir as the patron of this seminar or the colloquium has given us a boost for conducting the colloquium. We understand the presence and the cooperation received from national and international academia and researchers as distinguished speakers, and significantly enrich the value of the colloquium, and we hope that their deliberations and discussions will certainly help a lot to all the cross sections of the Indian and overseas participants. Thanks indeed. With these words, I would like to hand over the floor to my colleague, Mr. Joy Mukherjee, for the opening session. Over to Joy. Okay. Hello. Hello. Okay. Until Professor Joy would just join us. I would like to just say that the genesis of this seminar was actually to impart whatever type of education system, online system that we can impart to the students. And in this, we have got inspiration from our Honorable Vice Chancellor and Honorable Registrar, and also the professor in charge of the Dinhata College to go ahead, and they have given us a blanket approval for that. For that, we are very, very much thankful to them. And I think I must mention that all the resource persons, right from America, Sri Lanka, Nepal and China, and also from Singapore, they have joined us. And moreover, there is one more gentleman, resource person of national scientist from Botany Institute of Paleobotany in India, and we have received a tremendous cooperation and help from them. And I do hope that their deliberations in today's and tomorrow's this webinar would highlight everything on their part, especially in their research findings and also the education system in their areas to the students and also the faculties of our areas. Is Mr. Joy is there? Could you please join, Joy? Hello. Okay, okay. I have given an information that we have got some internet problem in our in college room. So shall we just be starting because we have taken a time from our honorable vice chancellor, our honorable registrar sir. So on behalf of Dinhata College, since our teacher in charge is under the weather today, therefore, I have been given a charge to explain on all behalf, and also Joy will be joining, and Devashish sir will also be joining, sir. And on behalf of this Colloquium committee, the organizing committee, and also on behalf of the Dinhata College, I extend my warm welcome to our honorable vice chancellor, sir, honorable vice chancellor, sir, and also registrar sir also had given us their time, valuable time to join in this webinar. And I see many more faces, known faces from USA and also even from Africa, that is Kenya, and also Nepal and China, and also from Bangladesh and all. So to all of you, and also my other speakers, honorable speakers from BSIP, they have already joined. And to all of you, I just extend my hearty welcome to this colloquium. And possibly just after the inaugural program, we will be starting our technical programs. And I just on behalf of Dinhata College request our honorable vice chancellor sir to just formally inaugurate this international colloquium, sir. So over to you, sir.
Thank you, Professor Thapa. At the very outset, I express my sincere thanks. Am I audible and visible?
Yeah, you're audible, sir.
Yeah, yes. At the very outset, I express my sincere thanks to the teacher in charge of Dinhata College and the organizers of this colloquium, and especially Professor Thapa, who has taken initiative to make this colloquium a grand success. And they have chosen a beautiful topic. The topic that has been chosen by the colloquium community, it is the science and society and the institutional approach. So far my knowledge is concerned and I have heard. Virtually what happens now, that is both the concepts, that is society and science, it changes over time. And when society demands something, science wants to put it, wants to give it. For example, right now we are facing a pandemic situation throughout the world, and we are seeing that in every corner of this globe, all the scientists, they are doing something in order to protect, in order to come out of this situation. That is, some scientists, they are engaged in discovering the vaccine. Some other also they are trying to discovering the medicines also. Then that is when society demands something, whether it is in the medical field or in other field, when society needs, when society feels, science comes out and science comes forward to help this, to solve this problem again and again. And here also one part of institution is there, and institution as because in especially higher institution. For example, here we can cite the example of Oxford University. The Oxford University, who has taken initiative to discover the first vaccine in this world for this COVID-19 pandemic. And this is the institution that has come forward to help the society, to come out of this problem, big problem. Like that, in India also, all the universities, they are also doing something to get something. For example, our Indian Institute of Technology, Colloquium, they have invented some machines also regarding sanitizations and for testing of COVID-19. Like that, all the institutions, they are all trying to, even, for example, our university also, they have come forward to help this situation. They are doing something. Our machine is helping the Cooch Behar district for detecting the COVID-19 cases. Our university also has come forward. That is, all these things establish one thing, that when society demands, when society needs, science comes forward, institution comes forward, and they are trying to help the society. This is the essence, and this is the quintessence of this today's colloquium. I am sure so many scholars all over the globe who have come forward here, and on behalf of this university, I express my sincere thanks for organizing this colloquium. Here, my colleague, our registrar, Dr. Abdul Qadir Safili, he is also here. He will say something, and also experts, panel of experts who are here and who already have joined. On behalf of our university, I express my sincere thanks for cooperating, for making this colloquium a grand success. And I also express sincere thanks that the organizers, they have given me opportunity to say a few words in this beautiful colloquium. Thank you very much. Over to Professor Thapa. Thank you.
Thank you very much, sir. Your blessing words are very, very boon for us. And then whatever the blanket approval and the blessings you have given us from the university, we are quite indebted to us today, all this. Okay. And then now I would like to request our registrar, sir, to have his views as the patron of this colloquium. Over to registrar, sir.
Am I audible?
Yes, sir. Yes, sir.
Thank you, Professor Thapa. Good morning to our Honorable Vice Chancellors, all the dignitaries, scholars, and participants in this international webinar being organized by Dinhata College in collaboration with the Department of Botany, Cooch Behar, Panchanan, Barma University. Hope you all are doing well. At the outset, I acknowledge that this is not my subject. However, I would like to share my personal two views emanated from my practical experience. Number one, in the northeastern part of India, and specifically in North Bengal of West Bengal, large section of native people use various plants for their treatment in various diseases, and in number of cases the results are positive, but unfortunately there is no scientific explanation or system of preservation of the treatment system. So I think there are ample of avenues for the scholars and researchers to explore the importance of plants and in the field of medical sciences. Number two, the role of botanist. We expect a very pivotal role for the protection from pollution in this type of COVID-19. We have seen that the positive result, at least in the field of pollution. It has decreased the pollution. So botanists can play, and they are playing, and we expect the vital role to play for the awareness campaigning for the sustainable development for our future generation. I hope this type of webinar will award the people, and some positive way out must be find out to the young, for the young generation and for the scholars, how to protect the environment and the ecological balances can be established, keeping in view the development that is sustainable development for our future generations. With these few words, I hope this webinar will be a grand success. Once again, thank you to the organizers for giving me this opportunity to share few words. Thank you.
Thank you so much, sir. Your words are also a boon for us, and also that gives us the inspiration to move ahead in near future in this COVID-affected situations and like that. And I do hope that these online activities and other things, whatever we could do other than the face-to-face teaching, would be the only tool during this pandemic effect towards the teaching learning processes. Okay, and then I would like to express my thanks to Honorable Vice Chancellor sir and also Honorable Registrar sir for their time given to us, and also for these deliberations and the blessing words. And since this short inauguration session or the opening session is likely to be at end, and I would like to hand over my mic to Joy Mukherjee sir for the conduction of this seminar or the colloquium. But before that, I would like to ask, or I would like to request our Honorable Vice Chancellor sir to kindly formally inaugurate this colloquium open. May I request our Vice Chancellor?
Thank you, Professor Thapa. Thank you, Professor Thapa. I hereby declare the colloquium open. Thank you.
Okay, thank you, sir. Thank you so much for your presence, sir. Joy, sir, would you like to just add something?
Thank you. May I leave? Okay, thank you.
Okay, thank you, sir.
Professor Thapa, may
Yes,
I leave now?
Our pleasure, sir, that you all are here today, and so whenever you have time, you can join, sir. We will be quite, should I say, as we will be just indebted, and also we will be blessed more on your presence, sir. So all of us, on behalf of the participants, I request you, both of you, sir, that if possible, you can just join in any time during our sessions, technical sessions, sir. Thank you, sir. Thank you so much, sir.
Thank you.
Thank you so much. Hello. Joy, sir, are you there?
Yeah, I'm here.
Okay, can you just take the charge now, just for this thing, before the technical session, whatever things that we have to just explain or like that to the audience. Hello.
Yeah.
Yeah.
Thank you, Honorable VC sir.
Yeah.
In the capacity of the chief patron of this international colloquium, as well as the Honorable Vice Chancellor of our affiliating university, that is Cooch Panchanan Barma University, you have spoken and delivered us a very inspiring speech, and also our Honorable Registrar sir, who is the patron of this Colloquium, and we, on behalf of Dinhata College and the organizing committee of this international colloquium, we extend our heartfelt thanks to both of you. Due to some technical problem, there was some hiccups at the beginning of this program, though we have overcome it. Now we'd move directly to the main session. Now I'd request Dr. Debashis Das, the Associate Professor of Chemistry and Chairman of Internal Quality Assurance Cell of Dinhata College, to deliver his.
Hello, Joy, sir.
Yeah.
Hello.
Yeah.
I would like to add something just before Devashish sir speaks. Okay.
Okay. Dr. Kishore Kumar Thapa will be speaking on behalf of the organizing committee. Kishore sir, mic is yours, sir.
Okay. Thank you. Thank you. In today's colloquium, two days colloquium, the first day, we have three scientists from two from abroad and one from okay, all three from abroad. Okay, the first one is Dr. Rna Saha, second one is Dr. Michael Stanley Baker, and the third one is Dr. Mudita Kasun Migakumbara from Sri Lanka. For this, then after that we will be having one discussion session and also winding up. And just before for that, for all conducting all these things, I request our my colleague Dr. Debashis Das just to take over the charge and start with the introduction of the speakers. Thank you. Thank you all. Over to Debashis sir.
Okay. Thank you. Am I audible? Yes, am I audible?
Yes.
Okay. Okay. Thank you. Due to some problem, technical problem actually, we are unable to join in from the very first session. Okay. Now, I introduced Dr. Rna Saha. Before that, it is my great pleasure to introduce the young scientist and very well known to most of us, our first speaker, Dr. Rna Saha. Dr. Saha is presently working as an assistant project scientist at Chemical and Biomolecular Engineering, University of California, Los Angeles, USA. He completed his BSc in chemistry from Siliguri College under the University of North Bengal and MSc in biotechnology from Jadavpur University. He was awarded PhD degree in biochemistry from SN Bose National Center for Basic Sciences, India, after which he went at the University of California for his postdoctoral work in the year 2014. He has worked intensively on the effect of minerals on RNA structure and function, RNA activity inside vesicle protoshell, and UV radiations on RNA aptamers. Interestingly, it is stated that behind this pandemic crisis, there is also a RNA virus. He has published a good number of papers in various well-reputed journals, and some of them are Chem Biochem, Annual Review Biophysics, Current Biology, and notably the Nature Communication also. In the last year, his research work has been cited in the cover feature of Chem Biochem. With these words, I would like to invite Dr. Saha to start his presentation. Over to Dr. Saha.
Thank you very much to the organizing committee for giving me this opportunity to talk on my research. I'm very much glad being part of the colloquium. So today I'm going to tell you about the time on the planet that we call RNA world. So let me start the presentation. Okay, is it visible, the slides?
Yes, yes, yes.
Okay.
It is
So
visible.
yeah, so
Okay, no problem.
my plan is today to talk about RNA world. So basically, so this is the this is the time before there was any DNA, and before there was any protein being present in the planet. So there is a very good collection of data that suggests that biology came from RNA, and then gradually other DNA and protein world gradually came up. So before I go into further details, I would like to tell you what is RNA molecule and how does it differ from DNA. So we mostly all know, like DNA. DNA is our hereditary molecule. It acts as an genetic information carrier. And if you look at the left side of this slide, you'll see like DNA is composed of polynucleotide chains, which is nothing but a polymer of nucleotide molecules, and it is double-stranded. Now, RNA is almost identical to DNA, but the important difference is RNA is mostly single-stranded, whereas DNA is double-stranded. Moreover, there is also some differences in the nitrogen-containing bases in between DNA and RNA. Both has adenine, guanine, and cytosine, but it is uracil for RNA and it is thymine for DNA. Okay, but these are like very minor differences. The most important difference between DNA and RNA is the presence of oxygen. So if you look at the right side, which is the RNA chain, you'll see at carbon 2 position, as I'm pointing by my cursor, like you see there is oxygen. There is oxygen present, okay, which is not actually present in DNA molecule. So this is, I mean, the oxygen is absent in the, so the oxygen is absent in the DNA, in the sugar moiety of DNA. And very interestingly and very significantly, the presence of this oxygen has huge impact on the biology. Because of the lack of this oxygen at the carbon 2 position, DNA is much more stable, approximately I would say 1,000,000 times more stable compared to RNA. And this has important consequences on the hereditary, on the consequences on genetics. Like because of the presence of absence of oxygen, DNA is much more stable, and that's why we see like in today's world, like DNA acts as an information carrier. Okay, but RNA is much more less stable, so it degrades very fast, so it doesn't act as an information carrier, genetic information carrier. So with this background, so if you look at the central dogma of molecular biology, you'll see information transfer starts from DNA. DNA codes for like DNA information transfer goes from DNA to RNA through transcription process, and then the protein synthesis takes place in the very complex machinery of cell called ribosome, and this process is called translation. But this process, although but there is a very, like, this process is very much complicated there, and actually this complicacy can be like, if we take more close look, you will see like actually what happens, like although it looks very simple, but this process is very much self dependent. Like each step of the process is very much dependent on the other factor. So if you look the replication of DNA, which is the copying of DNA molecule, it depends on other protein molecules. Okay. Now you consider the transcription of DNA to RNA. It requires DNA because there is the genetic information is stored there, but it also requires protein molecule like various RNA polymerase. And finally, the synthesis of protein molecule, which occurs on ribosome molecule, is nothing but a complex of RNA and protein, as I already stated. So in the context of origin of life, people are very much puzzled to see how this interdependent machinery could actually evolve from simple chemical system during the origin of life, during primitive Earth atmosphere, because you'll see like every step is very much dependent on the other. So this is actually a puzzle, and this paradox is well known as the chicken and egg paradox of origin of life, like which basically wonders what came first: is it the DNA or the protein molecule? Now, very interestingly, the answer to this puzzle actually came from the discovery of ribozymes in 1980s. Two famous biologists, I would say RNA biologists, Professor Sid Altman and Tom Check, they noticed a very interesting phenomena. They reported that like protein enzymes, like we know, like protein in our cell, biological cell, protein acts as an enzyme. But they claim that like not only protein, RNA can also act as an enzyme molecule. Okay, Professor Altman from Yale, he noticed that like there is a RNA molecule which is called RNSP. So this actually can look for other RNA molecule, and it can cut it. So it can cut the other RNA molecule, and Professor Altman noticed that this cutting event is billion times faster when RNSP is present. Like you can see, like tRNA is its target, and the cutting of this tRNA is billion times faster when RNSP is present. So this is one example of RNA molecule acting as an enzyme called ribozyme. There is, and then side by side, Professor Check at the same time, at the same year, also noticed that mRNA maturation of mRNA, which is basically also a cutting event called transesterification reaction, which basically joins two exons to form the mature mRNA, can also be self-catalyzed. So this intron can be self-catalyzed by its own enzymatic activity. So these are the two famous historical example which suggested that RNA can act as an enzyme, and we call these ribozyme molecules. And this discovery actually got them Nobel Prize in 1989 in chemistry. Okay, so moving on, there are other examples of RNA molecule where we can see like RNA can also act as an enzyme molecule. The classic example is our ribosome molecule. As I already mentioned, it's a complex of RNA and protein. And if you look the central part, the red part, where it's called the ribosomal peptidal transferase reaction center. So this is the center region where the translation of RNA, the formation of peptide bond actually takes place. And very interestingly, people found that the central region is actually nothing but an RNA molecule, an RNA enzyme molecule, which actually catalyzes this peptide bond formation. So after this ample evidence, we can now conclude. We can say like RNA can act as an enzyme like protein molecules. But now a very important question with respect to origin of life scenario is like, how did functional RNA evolve? Like how did this functionality evolve through evolution? Okay, so in this connection, this is the timeline to the early history of life on Earth. If you see, like around 4.5 billion years ago, Earth formed. Then gradually, Earth atmosphere became cooled down, and like stable hydrosphere formed on the Earth's crust. And then around 4.2 billion years ago, due to some environmental condition or possible environmental condition, we got to see prebiotic chemistry. So we got to see like building life, nucleotides, sugar molecules, and everything it formed. I don't know exact, like some environmental condition actually helped the formation of this prebiotic chemistry. And then what happened, like at some point, like around 4 billion years ago, some RNA molecule, like RNA-like molecule, we call like pre-RNA world, actually evolved. Now, as the time like passes, like we found, like at some point, like at around 3.8 billion years ago, like RNA world evolved. Like RNA world, I mean like functional RNA molecule evolved. And then eventually we DNA and protein like came, and around 3.6 billion years ago, and then finally diversification of life started. In this context, a very important point would be like a very important question will be how this pre-RNA world actually evolved to form more complex functional RNA molecule that we currently or presently see. So there could be multiple factors for this event, like for this one possible condition, I think would be like, would be the environmental condition, like maybe some environmental, because of some environmental condition, this actually helped the evolution of this complex RNA world. So in next few, I would like to some evidences, like where people would like to mention some possible environmental conditions, life, or in other words, RNA world could have evolved. Okay, so moving forward, so this is the famous this one iconic picture of deep sea. So this of like this type area, like region. Interestingly, you'd see like the surrounding area is full of life, steaming with life. Various kind of life is actually surrounded by in this condition environment. So the so this is an example of life at high temperature and pressure. Moving forward, so this is another example. This is the famous Rio Tinto in Spain. So very acidic environmental condition. pH is extremely low. It's around like pH is around 1.7 to 2.5. And people and you can see like this is full of life, both microbial, like prokaryotic, and also eukaryotic life. It's full, yeah. So this is an example. Okay, so extreme in like the this is famous hot. And this is, and you can also see here, like it is like there is also microbial life present. And so this is another example where we think like RNA world could have evolved, like life or RNA world could have evolved. Okay, so so this is a quite obvious I think like is biological cell, the confinement of biological cell, because if you model like most of the RNA and all biomolecules actually like are present within a confined condition of modern or biological cell membrane. We think like similar condition could be was present during the evolution of RNA. So basically, like organic chemists, like they actually were able to like extract various lipids from carbonaceous chondrite meteorites, and actually in aqueous condition they were able to form this kind of structures, and which is actually surrounded by lipid membranes, and this is very much similar to what we currently see as a biological cell. So people think like cell-like structures were actually were present, were actually possible during the primitive art in the origin of life. Excuse me. Okay, and we call these cell-like structures protocell. Okay, so and people actually like were actually able to see like this vesicle like structure, what you call protocell, can actually undergo it can grow, and it can also divide. Okay, at some point it can grow and divide. There are many ways. One important like one famous like I would say one condition like if you add lipid to these protocells or vesicles, it can undergo growth like you see, and then followed by that it can like structures, and when you mechanically you give mechanical pressure like shaking, and it can divide and give new daughter cells. So this is a classic example of biological cell division in possible, I would say not like possible cell condition where biological cell could have undergone growth and division, like protocell could have undergone growth and division. So motivated by these results, like this experimental findings, we decided to actually to check the activity of RNA. So we wanted to see like how this protocellular condition, like confinement condition of vesicle, could actually affect the functionality of RNA molecule. So, and we wanted to see like, so here is this is the picture like RNA, when a particular RNA molecule, like a functional RNA molecule is present within a confined condition. And we wanted to see how this activity of this RNA molecule is different when it is present outside the vesicle. Okay. So with this idea, this question in mind, we actually decided to study malachite green RNA aptamer. So these are basically synthetic RNA molecules artificially selected to bind the target molecule. So in this particular case, it is malachite green dye. So what happens, like when you add this dye to this particular RNA molecule called malachite green RNA aptamer, the fluorescence intensity of this dye changes hugely. Like you see, like from this picture, in free dye it has almost no fluorescence, but whenever the dye binds to the RNA, the fluorescence intensity increases significantly. Okay, so this is a binding signature, the activity signature. Like higher is the fluorescence upon binding, higher is the activity of the RNA molecule. So with this, with the help of this detection ability, we actually encapsulated this RNA molecule, this functional RNA molecule, malachite green aptamer, inside myristoylic acid vesicle. So myristoylic acid is a fatty acid molecule which was actually extracted from meteorite, as I mentioned. So this is prebiotically possible like lipid. And what we found very interestingly is that, so we actually monitored the fluorescence, like binding profile, basically the binding profile of this aptamer in the two conditions, as I mentioned, like inside vesicle as well as when it is present outside. And what we found interestingly is that you see like there is a change in the shift. There is a shift in the binding profile, towards lower concentration of the ligand, which basically suggested like inside vesicle, the binding activity is is much higher. Like at least the change is, you see, like is almost 4 times when it is present inside vesicle. The midpoint of the transition is much lower when it is present inside vesicle, which suggests like activity increases under vesicle confinement. So we wanted to, we are very much motivated by the result. Like it shows like inside vesicle activity is increasing, means it's helping the evolution of RNA. But we wanted to see what's going on, or like is it more general? So we actually tried different condition of the, like different other fatty acid composition of the vesicle, and we found like similar effect. Like here, like myristoyl alcohol was included in the fatty acid vesicle. Again, monomyristolene was taken. In all the condition, we got similar like effect, change in the vesicle activity, like 3 times decrease in like increase in activity. We found this effect to be true for in phospholipid vesicle condition as well. So phospholipids are the lipids that are present in our biological cell membrane, and here also we found similar activity increase. So the question that in the next stage we came to our mind actually is like what's going on, why this activity changing inside vesicle. Okay, so actually monitored the fluorescence of this aptamer with changing temperature. So what happens, like as you increase the temperature, the dye will the RNA molecule will unfold and dye will come out. So fluorescence will decrease. And here what we found very interestingly is that when the aptamer molecule is present inside vesicle, the blue data points, you'll see the temperature, melting temperature, the midpoint of this transition, it's much higher. It is like around 35 degrees centigrade when it is present inside vesicle. But when it is present outside, it's much lower, like around say 15 degrees centigrade. So these data suggested like due to encapsulation, RNA molecule is becoming much more stable. The local structure is much more stable. Okay, because yeah, the dye is coming out. So everything what we are seeing now is the local structural change. So we wanted to study this in further detail. So we actually did a shape assay. So shape is something like is totally dependent on the principle of reverse transcription. So I think reverse transcription is something that most of you are kindly aware of. Like it's actually been used to detect COVID-19 infection in patients. So this is the reverse shape assay is also based on the reverse transcription assay. And basically what happens, like if there is any, with some acylating agent, we actually modify the RNA molecule, and depending on the flexibility of the RNA, the RNA molecule is acylated. And when if you do reverse transcription, the reverse transcription is stalled, and the band intensity actually helps you determine like how the local structure of RNA in each base is modified. So this is the kind of the short story of this assay. And what we actually monitored these 2 bases, and 830 and 831. And what we found very interestingly is that when the RNA molecule is present inside vesicle, the binding transition is happens much at lower concentration of the dye then compared to when it is present outside. So this data again indicated us that like it's actually confirmed as like there is local structural change when encapsulation due to encapsulation of the RNA aptamer molecule. Okay, so with this, we actually wanted to know what is going on. So we did some theoretical calculation, and actually it's based on some principle. So it's like we all know like RNA is a biopolymer. So you can consider it as a string or a chain. So it's like multiple segments which are connected by joints, as you can see here. So multiple segments. So and then what you can imagine, like this like protein, like RNA could have 2 structure. It could be either very much compact or folded structure, or it could be unfolded or extended structure. Now what's going on, like whenever you apply some confinement, like vesicle confinement, you will disfavor the extended structure because it has limited space within, like vesicle has limited space where RNA can be encapsulated. So it disfavors the extended structure. Like extended structure is this, and it favors the folded structure. And because of that, like we see like actually RNA is much, when it is present inside vesicle, RNA is much more compact. Its melting temperature is higher and it's much more stable, and that's why it can bind its ligand, and activity is much higher. So to put this in another word, we think like vesicle encapsulation and is actually acted as a chaperone for RNA through confinement effect. So most of you already know, like in our biological cell, proteins could be unfolded or it could be folded, and the misfolding of protein can be actually overcome by other proteins which are called chaperones. And we think like vesicle in the prebiotic context, vesicle acted as chaperone for RNA. It disfavored this unfolded structure without affecting the folded structure, and this disfavoring of this unfolded structure actually gives relative stabilization of the folded structure, and that's why like RNA is much more stable inside vesicle and activity is much higher. So we can say like in word, like we can say like vesicle encapsulation was actually beneficial for the evolution of RNA enzymes. Okay. So my my final research finding for today's talk. So so we all know like UV flux at Earth's surface like 9 billion years ago was 2 orders higher. Now there is a very common misconception like that solar UV light is lethally bad. We know we have here like UV light is bad for our biological cell, but that is not always true. People actually found that UV light could be beneficial for prebiotic synthesis of molecular building blocks. So this is an example. Like if you start with hydrogen cyanide, you then shine UV light on hydrogen cyanide, and as the reaction progresses, you see like you can end up getting 2 to 3 carbon sugars. You can actually form pyrimidine ribonucleotides. Even lipid precursors like this can be formed. And not only that, various amino acids, like 10 total amino acids, could actually be formed from this simple chemistry, from starting with hydrogen cyanide in presence of UV light. So motivated by this, the potentiality of this UV light for in helping the synthesis of molecular building blocks of life, I mean lipids and RNA and like nucleotides and sugars, we actually wanted to study how UV affects the functionality and replicability of RNA. So yeah, so in this study, in addition to the malachite green aptamer that I already introduced to you, we plan to choose another aptamer molecule which is called spinach. It has similar characteristics, but it binds a different dye called DAPGBI1T. And what happens is the same, like when the dye binds, the fluorescence of the RNA enzyme is significantly enhanced. So that's why, like, since enhanced, these are called fluorescence RNA aptamers. So we chose these, and we actually monitored the activity of this RNA at various condition of like at various exposure of UV. And what we found interesting is like with time, like fluorescence of these 2 aptamer decreases. So that means UV is actually is degrading this RNA through some kind of damage. But what is very interesting is like the damage rate is different for these 2 RNA. For malachite green aptamer, which is blue here, you see like the damage is much higher. The rate of damage is much higher, and also the extent of damage is also much higher. But in case of spinach, it is rate of damage is low compared to malachite green. The rate is for spinach, and also the extent of damage is much less, like at least 20% less compared to malachite green, because at the saturation of damage, you still see some fluorescence from spinach. Okay, so we wanted to see what's going on, like what is the structural modification of the RNA molecule through UV damage. So we actually monitored the melting temperature of these two of the malachite green aptamer, and what we very interestingly we found like the melting stability of the aptamer, malachite green aptamer decreases from 34 degrees centigrade to 12 degrees centigrade in aqueous condition, and we confirmed this destabilization of the RNA by running some both native and urea gel, and we found like due to UV damage, it generates another new band in the DNA gel, which suggested like there is misfolding of the malachite aptamer due to UV damage. So next we wanted to see what's going on with spinach aptamer, and here we found there is no change in the melting temperature due to UV damage, and which is very much consistent with the result that we got with respect to rate of damage due to radiation. So we wanted to study this thing in further detail. So we actually plan to choose reverse transcription assay, which basically can tell like where the damage happened. So consider this is the RNA molecule, and suppose there is some damage due to UV, and now if you do reverse transcription, you want to copy this, basically you want to copy this RNA through reverse transcription assay in presence of some enzyme. What happened, like reverse transcription is stalled due to this formation of this adduct. So you'll see various bands in the gel, and through running sequencing, you can see where the damage is actually happening. And this is also like, yeah, so with this, with the help of this technique, we were actually able to see like where the damage happened in the RNA, and we very interestingly found that damage is much more in the binding pocket of the RNA. Like binding pocket, I mean where the ligand molecule is actually bound to the RNA, and we found like the damage is happening most in the binding pocket compared to the folded region. Folded region means the stem region of the RNA, where like bases are much more hydrogen bonded, like here and here. The damage you see is much less, but its damage is more in the binding pocket. We quantified this data through another way. We monitored the UV reactivity of this RNA with time, and here also we found like the damage is very much the rate of damage is much more prone in the binding pocket of the RNA compared to where it is like much more hydrogen bonded, or it's much more folded. So this is much more folded structure compared to this where the dye actually binds, and it is much more prone to UV degradation, UV damage. Okay, so coming to the summary of this second part of the talk. So we found like UV exposure actually creates chemical lesion in RNA. We found this is a sequence dependent manner, because if you remember, I showed you like for one malachite green aptamer, the rate of damage is much higher. However, for spinach it's much lower. So we think it's the damage of RNA by UV is a sequence dependent thing, and we think like it could, multiple factors could be responsible. We don't know the exact reason for the different rate of damage. It could be that lesion formation is mutually exclusive. It could be that like formation of UV and damage formation in one region of the base, it mutually excludes the formation of the damage in the other region. So that could be one reason, like spinach is much less prone to damage. There could be some self-repair system that's going on. We don't know that could actually help spinach to overcome the UV damage, and also the structure of the binding pocket. Like binding pocket structure could be different for spinach, and that's the or could be one of the reason, like rate of damage could be different, like, and rate of damage as well as extent of damage could be different for this RNA. Okay, we also saw like UV modification. It actually stalls the copying ability of RNA. So we think like UV modifies the templating ability of RNA. And also we found like increased susceptibility of damage in the binding pocket, because compared to stem, like if you remember, I showed you like it's more, the damage is much more prone in the binding pocket compared to the folded region. So we conclude that like well-folded functional RNA was much more favored during evolution in the context of UV damage. Okay, So, this is the we think this work highlights the importance of UV exposure as one of the important selective pressure during the origin of life. Okay, so finally, I would so this is the campus, like University of California campus, like where I did my research. This two research, my postdoctoral research. This is nicely located in by the Pacific Ocean. The whole this is the campus where it's very close to the Pacific Ocean. This is the famous Henry Gate of the university, and this is kind of overall look of the campus. This is our department. Our lab was actually housed within this building. Okay, and so currently I'm now working in University of California, Los Angeles. This is the Chemical and Biomolecular Engineering department, where our lab is now located, and I'm hoping to continue my research here and hoping to get much more, more interesting results on origin of life. So with this, I'd like to acknowledge my whole lab, the Chen lab, also Professor Chen, Sam, who actually helped me in this project, in doing the series shape assay, and also our both UCSB and UCLA, NASA Astrobiology Unit for our funding, and also Simon's Foundation, and also Institute of Collaborative Biology, ICB. So with this, I'd like to thank everyone, and I'll be happy to answer any question if you have. Thank you.
Thank you, Ronojoy.
Yeah.
There are so many questions came in the chat box.
Okay.
And due to shortage of some time, I can choose some of them, some of the questions from it. First question is, why is the damage more prone in the binding pocket? Random or Darwinian selection?
Okay. Yeah, I think I think it's just look, as I mentioned, like I showed you, like the damage is less in the stem compared to the region where RNA, where the dye binds. We think like it's the folded structure of the RNA, like folded region of the RNA is less susceptible because of the hydrogen bonding and all, but the region where like dye binds is much more like unfolded, so possibility of damage is like much higher, like bases are more prone to damage. Yeah.
There is one more question asked Wangyal Bhutia. He said, can distress, our emotion, impact the genetics of telomere of a chromosome?
Can what? Sorry,
can distress, our emotion, impact the genetics of telomere of chromosome? Anything, some change from the behavior of us, whether there is any impact on the genetics of telomere of a chromosome? Like distress?
I don't think I'll be right person to answer on this. Yeah, like I like my knowledge is very much limited on the activity of telomere.
Actually, it is somewhat totally confined in the problem of chromosome, that is genetic code or something like this. I hope so. And basically, you have done work in the RNA and DNA also. And you have said in your presentation that is hybrid DNA. So one Dipayan Banerjee, he asked, what is hybrid DNA?
I didn't tell about. I think hybrid DNA means like it could be like DNA that is hydrogen bonded with another DNA strand.
Yeah.
Yeah, but I didn't work with DNA hybrids. Yeah.
Actually. Yeah.
So
And
basically, the DNA hybrid means like DNA strand, like if you have two DNA, like say two artificial DNA strands, and it's hydrogen
yeah.
bonded, that's
Yeah.
called DNA hybrid. Yeah.
That is DNA hybrid. That can
Yeah.
be said.
Yeah.
Okay, you have said one thing. This is my question actually. Prebiotic chemistry.
Mm-hmm.
You just say some words about the prebiotic chemistry.
So basically, like this is the chemistry, organic chem, basically some synthesis of biomolecule, like possible like building blocks of life, like organic synthesis of building blocks of life, say ribonucleotides or say sugars.
Yeah.
So those are the building blocks from which then say protein or say DNA or RNA actually was synthesized basically. So these are the polymers of those building blocks, right? So one group of people in the origin of life field, they work on the synthesis of those possible synthesis of those building blocks. So I think one possible way I showed in the slide, like where I showed like due to UV radiation, you start with hydrogen cyanide, you can gradually you can gradually synthesize most of the blocks, like building blocks, like say nucleotides, say sugars, 2 to 3 carbon sugars, say amino acids. So I mean like that, like so UV radiation is one possible condition in like as I mentioned, like the UV radiation intensity of UV radiation was much more higher in prebiotic chemistry, like because we didn't have our atmosphere was different, so intensity of radiation was different. So one possible organic synthesis of molecular building blocks could be like UV radiation. So there could be other ways as well. People are working on it.
Actually, what you have said, that means the synthesis of the biomolecules actually. And but my point of questions is that, is there any necessary conditions for prebiotic chemistry? That means if the reactions fulfill this type of environment, then we can say it is a prebiotic chemistry. That means synthesis that may be done in certain environment. So I ask this. I ask of this type of environments. Actually, what type of environment?
It's nothing is very much certain. So that's why we call it plausible condition. Like, so as I mentioned, like it could be various like it could be temperature, it could be high temperature, it could be high pressure, like then radiation could be one condition. So there could be multiple plausible conditions. People don't know, but we all think like some extreme condition that was present, and as I tried to show, like actually like life can still survive in those extreme conditions. So why not like RNA? So that's why we chose those conditions and see like how activity of RNA changes. So but nothing is very certain, but we just try. Like
that means
we see example of today's world. In today's world, we see like in what extreme condition life could survive, and then we think maybe it could be one of those conditions, and then why not see like how RNA behaves in those extreme conditions.
Okay. It's all right. That means you may choose this condition by trial and error method. So you see the current condition, like where life could survive, and then so basically some extreme condition, and then you try like how RNA behaves or other molecular building blocks behave. One more thing, you just say in a very nutshell. That means you said that that means the damage of RNA. I have just asked you one question, very simple question. That means what is the basic difference? That means a proper RNA and whenever it becomes damaged, mainly giving emphasis on the function of RNA. And what type of difference that was seen in a pure DNA and in a pure RNA and a damaged RNA?
What kind of difference?
In the function of RNA.
So yeah, so as I showed, like due to like base modification, we found like through reverse transcription, base is modified, like base is changed. And you see, like if you remember my slide, like I did reverse transcription, like copying of that strand, and that copying is stalled because of the damage. So that is one of the
Copying is
reasons.
stopped actually.
Yeah, copying
Yeah,
is
yeah,
stopped.
yeah.
Like you see,
Yeah,
that's why you
yeah,
see
yeah,
like
yeah,
random stop. Yeah.
I mean, that means that the activities of RNA be restricted by its such a damage, right?
Yeah. Yeah.
And damage means such a here by using some binding ligand.
No, so binding ligand, no. So the thing is,
Actually,
in my case, binding is the one signature. Like I chose like that RNA molecule which can bind a particular dye, fluorescent dye.
yeah,
But
fluorescent dye.
but the damage doesn't depend on the dye molecule or the ligand molecule. So
Does not depend on the ligand molecule.
no,
Well,
it may depend. Sometimes like we call like the structure of the RNA modifies due to binding.
yeah.
And this is called induced fit model. Like you remember, like induced fit modification of RNA or any biomolecule, like ligand binding modifies protein structure or RNA structure. So that is a different story. But when we did this, when actually performed this damage, like UV damage, we didn't take dye. So whatever you are seeing is not influenced by the binding of the dye.
Binding of the dye.
Yeah.
Okay,
You got my point? Like I mean, like we did the UV radiation instead of, like in absence of dye. So whatever damage you are seeing by in the copying chemistry is irrespective of the dye being present.
Yeah. Okay, another one more question is that apart from DNA, do you think some other entities that may have survived in the prehistoric period, from which the life may be in force?
Yeah. So people are working with other modified I think modified polynucleotides, I'd rather say. So like DNA, RNA. So these are like we now see in our modern biology, in modern biological world. But there could be other possible polynucleotides, like building blocks, where the building blocks could be different. Like I think one example is TNA. Like TNA is one like molecular building block which is possible. Like people are working with different those modified like polynucleotides, and seeing whether those polynucleotides could actually copy itself, as well as could act as a hereditary molecule. But basic thing is like for life, like DNA can copy itself. So like RNA. So sorry, I don't mean like DNA copy. Like one important like characteristics of RNA is that, as I mentioned, like RNA can copy itself. It can self replicate itself. Yeah. So people are working with modified RNA and trying to see like whether those modified polynucleotides can copy itself.
Okay. Yeah. Okay. There are again so many questions, but we are actually, due to lack of time, we will provide the email ID of Dr. Rna Shah attendees. If you have any further more questions, you can consult with him in his email. Okay.
Sure.
Now, thanks. Now, on behalf of the organizers of this colloquium, I convey my hearty thanks to Dr. Rna Shah to find time and enriched us for his lecture, The Emergence of RNA Functionality Under Potential Prebiotic Conditions. This talk illustrates the diversity of functions like proteins, leading to the hypothesis that during origin of life, there was an RNA world, from which the more complex functional DNA RNA protein world has evolved. It is surprising to know that the UV radiation, recently known as but at the early stage of this planet, these UV radiation are important for prebiotic synthesis of amino acids and ribonucleotides. We sincerely hope that you will do more fundamental and thought-provoking research in near future in this area that help us to know some extraordinary and interesting findings about revolutionary consequences and functions of RNA. Thank you, Dr. Shah.
Thank
over
you.
to Joy Mukherjee.
Thank you, Dr. Debashis Dash. Words of thanks. Now I would invite Dr. Manjil Gupta to introduce our next speaker. Manjil sir, the mic is yours.
Thank
Please
you.
take over.
Thank you, Joy sir. It is my honor to introduce our next speaker, Dr. Michael Stanley Baker. He is currently the Assistant Professor of Medical Humanities, History and LKC School of Medicine, Nanyang Technical University, Singapore. He is also the Vice President of Indian Association for the Study of Traditional Asian Medicine. He completed his BA in Philosophy and English from the University of East Anglia and MA in Chinese Religions from Indiana University, Bloomington. He was awarded PhD degree in Medical History from University College, London. He has published over 40 papers in different journals, book chapters, volumes, journal special editions, Prebiotic chapters. He has also contributed to many databases and datasets relating Chinese medicine. He has also received many research fundings and awards. This is just a very small fraction of his illustrious career. So I would like to invite Dr. Baker to deliver his talk. Over to you, Dr. Baker.
Just a minute, Manjil. Just before Dr. Baker takes up, Dr. Kishore Kumar Thapa wants to add a line. For the interruption.
It's okay. It's okay. Okay. Michael, are you okay, Michael? Hello?
Yeah, I'm here.
Okay. 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 Michael, you can proceed now.
Okay. Gotcha. Hi. So I should add a correction to my introduction. Thank you very much for the very kind introduction and for the opportunity to speak with you all today. But I'm not the Vice President of the Indian Association of the Study of Traditional Asian Medicine, but the International Association for the Study of Traditional Asian Medicine. There is an Indian chapter of ISM who are doing fantastic work, and in fact are an even larger body than the international one. So I just want to make that clear from the outset. So I'm very impressed by my predecessor, who's speaking so eloquently about the origins of life, DNA and RNA. I confess to be somewhat at a loss, because I was asked to not do the opposite and not give a scientific talk on detailed elements of my research, but rather to give a very general introduction to Chinese medicine. So for those of you in the audience who are expecting sort of high-level, postdoctoral science, I apologize in advance, but that's not what I'm going to be doing. It'll be just a general welcome mat to the field of Chinese medicine in general, and we'll start out by describing some very fundamental principles that emerged in the early history of Chinese medicine. Just to give you some context, all of the whatever academic accolades aside, my background in relation to this, I come to it initially as a clinician. I started the study of Chinese medicine, of the clinical practice of Chinese medicine in my 20s. I was just calculating yesterday, 20 years ago now, but very soon found that I became deeply interested in the historical background, the cultural traditions, and really went the way of medical history and medical humanities. So things that I see as the key points that needs to be addressed in today's use and spread of not traditional medicines generally is this issue of translation and transmission, and how do we knowledges with current day science, and the demands of increasingly regimented, increasingly based regiments, not only of bodies but of knowledge. And it was with great respect, actually, to the Chancellor who began the opening today by stating that people have really not been addressing COVID with scientific means. They've been using herbs and traditional methods. And I want to propose towards the end of my talk that actually I think we should look at science not as a synchronic thing, that there is only truth and falsity, and if it's been proven within the latest scientific methods, then it is true, and if it hasn't, then it is false. Science is actually a long-standing history, and in China, at least recorded history for at least 2,000 years. And that's what I'm going to continue to talk about now. So I'm going to now share my screen. Let me see. So excuse me, well, it may take me a little while to do this. Sure. Okay, so can you see my slides now? I think that you can.
Yes, yes.
Yeah? Okay. So there you can see the title properly. It's International Association, not the Indian Association, with great respect and love for ISM India. Okay, so it's often said that Chinese medicine is a 5,000-year-old tradition, and China itself is a 5,000 or longer-year-old cultural tradition. Be that as it may, recorded medical history and the medical systems that are still in use today do not date that long. They date back basically 2,000 years, and they sort of became formalized, and I'll show you a time slide in a second, about 2,000 years ago with the formation of the Chinese Empire as we've known it for the last 2-point-some thousand years. But without getting into a long durée history of China, I'll first introduce to you what I'll be discussing, which is initially 3 main concepts. That's yin and yang, the 5 agents, and qi. What is qi? I happen to be writing a paper on that question right now. And then I will discuss some of the treatments that are used. Very often, acupuncture and moxibustion are touted as the primary sort of significant Chinese modality. This is the one that marks Chinese medicine as distinct because we use needles. But in fact, herbs have always been and are currently the predominant use, the predominant therapy. But in addition to that, you also have exercise and diet. Then I'll discuss very briefly some methods of diagnosis and some simple disease theory. And then I'll conclude with some questions about Chinese as a modern medicine and questions of integration, particularly in relationship with COVID and how Chinese medicine has been responding. Okay, so the classical medical theory is grounded in this text here, which was compiled around the 1st century, I don't know if you can see my mouse, but around the 1st century CE, called the Yellow Emperor's Inner Classic, named after one of the founding emperors of China to give it the imprimatur of antiquity. it in fact gains its coalescence in the Han Dynasty, which is the first major long-term unified state, at which time you have a whole kind of standardization of knowledge and this very intimate relationship between the cosmos and the political scene, which you see reflected in many of the notions in Chinese medicine. This notion of the interior of the body being reflected in the exterior cosmos and vice versa is a predominant theme in there, and we'll see how that plays out in terms of disease ideas later on. Now, coming after that, around the year 200 CE roughly, are two important texts which are important to bear in mind. The lower one that you see there is called the Divine Husbandman's Pharmacopoeia, and that is basically a description of single drugs. And each drug is analyzed for some of its properties, its flavors, how to recognize it, where it grows. And this becomes a textual tradition that lasts for 1,500 years, or 1,300 up into the year. The last major edition was 1,500, and then you have now modern pharmacopoeia, which take quite a different form. The other text, however, is this treatise on cold damage, the Shanghai Lun, which, not unlike the situation today, emerged in a context of extreme epidemics. In fact, from the year 0 up through the year 400 or 500, you can see in Chinese history, historical documents, epidemics are described as appearing every 4 to 5 to 10 years in very high rapidity. And it was clearly a critical, had a critical impact on the consciousness of people at the time. How to escape disease, where was it heading? My presentation seems to have shifted somewhere, but anyway, I'll assume it's still working until I hear otherwise. So the Shanghai Lun, the treatise on cold damage, began to develop philosophies or methods for combining different herms together and how to recognize the progression of disease, whether through symptoms like sweating, through tightness in the neck, through vomiting, fever, etc., and how to identify it, track it, define it, and isolate the appropriate herbal combinations for that. This text circulated like many others until it didn't really stand out until the 11th century, when during the Song Dynasty, when the imperial state formed a medical academy, and this became the hallmark text for medical officers, literally who at that point became officials within the Song government. And they had to learn this text, and from that point on, at the same time, China also began printing texts. So you had broad circulation of knowledge, and all of these scholars studying this text to get into office and to gain position, and that resulted in an explosion of new recipe combination theories, which emerged over time. There were 4 main schools, which sort of continued on for the next 900 years until the present day. But the rise of that text was because of, again, epidemics that were taking over and affecting the economy of the Song Dynasty. And it was such a problem during the middle of the 11th century that they began to implement the Shanghai Lun as a primary response to it. So we see that the Chinese medical theory and herb combination theory has a long history of response and adaptation to epidemics. Okay. So what are some of the primary concepts that we need to gain grips with? Okay, we can ignore that. The first one, I'm sure you all recognize the symbol, is yin and yang. And there are many different descriptions of it and its implications and what it means. As you can see, there's a large, black, some people describe it as fish, heading upwards on the left side, and a white fish heading downwards on the right side. And then with these 2 eyes in the middle. And what this represents is, as yin, starting at the bottom here, the black sort of tail of the fish rises and expands upwards. It grows and becomes more powerful. At the end, when it is apex, it then transforms into yang, which is represented by the white dot. And then yin's power declines and yang emerges and becomes stronger and stronger. So we have this interactive dynamism that is being expressed by by the theories of yin and yang. And it's important to recognize this interactivity and this dynamism. So when people talk about the origins of the term as referring to the sunny side and the shady side of the mountains, you've got to remember that the sunny side and the shady side of mountains never stay sunny and shady all the way through the day. In fact, it's a constantly changing landscape. And it is the attempt to identify where one is as one standing in that stream, as it were, in the midst of changing events, that is the motivation behind the principle of yin and yang. So we, think about it from these very common doorway kind of terms as being masculine versus feminine, bright versus dark, warm versus cold, dynamic versus static. When you look at the components of the body, we have qi versus blood, and I'll get to qi in a moment. The power of ascending, rising is the yang, descending is the yin, and dryness is yang, and yin will be damp. So all of these conditions are malleable, changeable, and fluctuate. And the point of using yin and yang is to identify these transformations. Now, the other principle is the 5 agents. Now, you are no doubt familiar in Indian lore from Yoga Tantra and other disciplines of this principle, the 5 elements in Indian thought, right? Which at its most base is earth, and then water, and then rising to fire, and then becoming more refined to air, and then ether merges into the absolute. There's this ascending scale of refinement, right? As you approach the divine. At least certainly this is in this picture here, which I taken up the internet, which refers to the chakras here in the Sushumna Nadi, rising up the central column of the body. And these elements in Indian medicine, and I'm sure you can tell me much more, really are very important for Ayurvedic diagnostics. So earth and water forming kapha, pitta being the primary dominant in fire, and ether and air, air and fire becoming combining in vata. So this is this ascending grades of refinement in the cosmology in India is quite different from what's happening in Chinese medicine. So one, we have different agents rather than elements, they're really agents. So we have fire, earth, and water, but we have wood and metal. So that's one difference. But the other more important one is this cyclical relationship. The external circle that joins these 5 elements, five agents, is what's called the supporting cycle, as you can see in the graph here. So, or the generating cycle. So wood generates fire, of course. Fire, when it burns out, can then generate earth. Earth then generates metal deep down. When metal is produced and left outside on a cold, dewy morning, it will form water on it. And water can generate wood, of course, can give life. Now, that's one relationship of generation and support, but there's another one of control that is internal here. So you can see water controls fire, as we can imagine intuitively, right? Water, of course, puts out fire, and fire can melt metal, so it controls metal, and metal controls wood, because a wood, an axe can chop a tree, and wood can control earth in the way that the roots of a tree can form the shape of a riverbank. And earth, the banks of the earth can control water. So there's this tension between supporting and generating that is at the heart of five-phase imagination. And that's important when we think about, it's important for thinking about physiology and the relationship between the different organs and how they function to support one another. But not only the organs and their relationship to each other, but also the human body within the larger cosmos, within the seasons. Because each of these phases, each of these agents corresponds to particular seasons. Let me first describe to you the internal organs that correspond here. So we have wood at the top, which corresponds to the liver and the gallbladder. If you think of the rising sap during the springtime, this is like the function of the liver and the gallbladder to spread qi upwards and around the body. Fire is associated with the heart, fast and moves quickly, and it's related to the intellect, the quickness of the mind, which resides in the heart in Chinese thought. Soil, on the other hand, is related to the spleen and the stomach, that capacity to absorb, to digest, that slow assimilation process. That's part of the kind of fundamentals of soil. And Jin, or the metal agent, corresponds to the lungs, which has a descending function. We're moving from yang to yin, and although I haven't explained it yet, we're also moving through the seasons. So the wood metal corresponds to spring, the fire corresponds to the summer, the soil corresponds to the late summer, early autumn, and the metal corresponds to the full autumn. And the lungs have this descending function which return fluids back down to the body. And then the water element corresponds to the kidney, which stores and processes water and holds deep within, particularly, and corresponds to the wintertime. So these correspondences have enabled people to, now we can look at it, we can think about it on the one hand and say, well, this isn't exactly science creation, this isn't a test case laboratory science, but it is a way in which early Chinese during the Han Dynasty and earlier observed natural phenomena around, saw correspondences that were happening at the same time and that were related in some way. Their relationships not necessarily very thickly explained, yet they definitely occur at the same time. So when people have specific kinds of diseases in the winter, in the spring, in the summer, in the fall, these are the kinds of knowledge and insights that the 5 agents theory is trying to capture. So I want to draw your attention. I've talked about the organs at the top, and I've talked about the seasons. The environmental factors are also important, and these actually constitute the classical pathogens and how Chinese medicine understands disease to function. So when someone has a fever, it might not necessarily be described as a bacterial infection. It would rather be described as invading wind that has penetrated the outer layers of the body and entered the body's system and then caused it to heat up in response to drive the wind back out. So wind has multiple meanings, but it's predominantly related to spring and to the wood pattern agent. Heat in the body, of course, corresponds to fire. Dampness corresponds to earth. I talked about that as the ability of the stomach and the spleen to absorb. But of course, when too much water floods a plain, the soil becomes bogged down. So dampness is related to soil here. And the metal element corresponds to dryness, excessive dryness. I'm thinking of Rajasthan as a very dry, arid area. And the diseases that dryness often affects are the lungs, and autumn is often a very dry time. And of course, cold corresponds to winter. So you have this large, this small system of 5 agents aligning a whole cosmology of the internal organs, the seasons, and also the different kinds of weather patterns. Further on, there are also, you can see down there are emotions, there are tastes, there are colors, there are sounds. The tastes are quite important for understanding the function of herbs. Most herbs are often, when you train in a Chinese medical school, you'll be asked to taste the raw herbs and learn to discern from the taste of the herbs what their function should be. You should be able to kind of get a sense or a feel for how it enters the body. And the taste is the first contact with that. And in fact, a lot of herbal recipes where you're combining multiple herbs together is not unaligned with cooking, the combination of different spices and flavors together. The other important aspect from this table I want to draw to your attention is the emotions, because these also correspond to the different organs. So someone who is excessively angry will be described as having a liver problem, possibly heat in the liver. And from a brief time I spent in India, I recall people describing this in the same way in Ayurveda. Joy, excessive joy, this is considered a problem of the heart, of the mind. When people are left, go into deliriums of ecstatic transport and can't control themselves, this is a problem of excessive joy and considered a problem of the heart, which, remember, is the seat of the mind. So these other organs, these other emotional elements, are in a sense the internal cosmos that is imagined in the Chinese body. Okay, so I've talked about yin and yang. I've talked about 5 phases. The third important one is qi, and I'm sure you've heard of the term prana. In fact, this term functions in many similar ways, but it is dealt with and perceived in China in culturally different ways. It's related to the breath, to wind, and it's understood to be able to move through the body, to nourish the body, and control its many functions. By the time the classical system formed in the 1st century of the 1st millennium, this is when we start to have the first really full descriptions of the channels, as you see in the picture here, which are bilateral, so they function the same across both sides of the body. There are about 360, possibly more, different acupuncture points along these channels, which flow along the surface of the body, but also flow interior to the body as well, as you can see in these diagrams. This is an image of the stomach channel, which, as you can see, starts up on the head and then comes down the cheek, down the neck, and along through the nipple and along the belly and down the frontal surface of the leg, and down to the second toe. However, you can also see this red line, which is an internal channel that goes through and connects to the internal organ. So although you're manipulating qi along the channel, you're actually affecting the organ. So a lot of treatment of the internal organs is done through working on the surface of the body. We should also, I want to pause too, and before I go on to talk about organ function, which I believe I have listed further here. No, actually, let's talk about organ function here. So remember when I was talking about emotions and the agents and the other aspects corresponding to particular organs. We really got to think about organs in a very different way. Some people describe them as orbs, as locuses of activity. So the stomach, for example, and particularly the spleen, they're composite systems. You see a spleen diagram, the spleen channel on the right-hand side. So these are 2 paired organs, the spleen and the stomach. The stomach is considered the yang organ, and it's called the fu. It's considered a hollow organ. And the more important and fundamental one is the yin organ, which is the spleen, or it's called the zang in Chinese. And together they form this system, the stomach and spleen, of digestion, governing mentation, sort of mental absorption, but also produce qi within the body. They also process grains and then send out nutrients, both in the form of blood and in the form of what's called grain qi, up to the body, up to the lungs, where it circulates, and the lungs then circulate that, transform that qi, and then send it down through the rest of the body as the kind of nutritive qi that we see circulating through the different channels, which is what you are dealing with when you're needling. So the correspondences that come from the 5-phase system produce different kinds of observations. So if you want to, you can do diagnosis, for example, of the 5 organs just by looking at the face. The eyes reflect the liver, the tongue, which speaks the mind, reflects the state of the heart, the nose reflects the lungs.
Dr. Stanley, please unmute yourself. Dr. Stanley, please unmute yourself. You are not audible. Dr. Stanley, please unmute yourself.
Okay. How long have I
Thank
been?
you, sir.
How long
Thank
have
you,
I been muted for?
Pardon me, sir.
How long have I been muted for?
A few seconds.
A few
Not
seconds.
more than that.
Okay.
Yeah,
Okay.
yeah.
Good.
Thank you, sir.
Okay. Thanks for the warning. Okay. So I was just describing how the complexions of the 5 agents can reflect the state of the 5 organs. And you can see these on the face, and you can also see them around the body as well. Okay. So when we talk about herbs, what I'm showing you here is a category of different kinds of functions and the ways in which these herbs treat the body. So this is from Bensky and Gamble's Materia Medica, which is taught in Chinese medical schools in the US and in Europe today. And you can see that they operate in ways to clear heat, release the exterior. Now, what does release the exterior mean? Many of these terms only make sense when you get a full sense of how diseases and the body are conceived. But releasing the exterior, remember I talked about how wind, heat joins, how wind can enter the body, penetrate the surface layer, and then cause the body to warm up and produce fevers in order to drive out heat. But if you have a fever and you don't sweat, you're not going to get that heat out of you. So releasing the exteriors and
Once again, sir, you have got mute. Once again, sir. Please unmute yourself, sir. Thank you. Thank you. Done. Done. Done.
Seems
Thank
to
you,
be
sir.
happening spontaneously. I'm not touching the computer, so I'm not sure why it's
Maybe, sir.
doing that. It might be happening on a timer. Okay, so then you have a few sets of herbs which would deal with dampness, and you may know of the similar symptom of kapha, right? Of dampness and stagnation in Ayurveda. So you can drain it out through diuretics. You can expel wind damp through sweating through the pores. You can transform phlegm in the lungs and coughing. And there's also things which relieve food stagnation when your digestion is poor. Remember how I talked about just earlier the critical role of the stomach in generating qi for the body. So when you want to heal a patient that's very deficient, you really have to work on their digestion so that they're getting nutrients from their food. To regulate the qi, either to make it flow or to cause it to become more even. Regulating the blood is critical, particularly for women's medicine. From the 7th century onwards, gynecology starts to focus very deeply on the texture and quality of the blood, particularly in the menses, and it becomes an identifying feature for women's conditions and problems, and they'll be diagnosed through very refined descriptions of their menses. So in order to treat anything from infertility to dysmenorrhea, you will use blood regulating herbs. There are herbs which tonify, that is, they strengthen the body, they augment the qi. Those which stabilize and bind if you have lots of secretions or if you have ulcerations, prolapses. There are psychological drugs which calm the spirit. And for those with fits, with tremors, with tendon problems that cause shaking, these are resolved through herbs for winds and tremors. And then there are herbs that work on are more empirical. They expel parasites, they treat skin problems, and those are rather simple and direct, and they don't involve a great deal of clinical medical theory. Okay, that was a dense slide. Let me make sure you have still got me on audio. Okay. So thanks for bearing with me. So that's the herbal medicine is the primary treatment even to today, but the one that has made the strongest impression in the modern world has been acupuncture and moxibustion. And as I've shown you with the channel system, you get a sense for how these needles, when you insert them into particular points in the body, these points lie along the channels, and you're actually affecting things quite distant, in distant places. So one could insert a needle, say here, which appears to be in the middle back, in order to strengthen the spleen and the stomach. This could be a treatment for digestion. Similarly, when there's an absence of qi, or there's from when people are weak or they have deficiency, you can apply heat with moxibustion, and that's the picture you see on the right, where the practitioner is burning a cigar of artemisia or mugwort and warming up the points. Okay, now another important element of Chinese medicine is exercise and diet. The qi can not only be moved by acupuncture or by herbs, but also just through plain exercise. And these exercises don't only move the exterior of the body, there are also internal components where you imagine and feel the qi circulating through the limbs and through the different organs of the body, taking different colors, etc. While these circulated through religious environments, early Taoist sects would use these by the Sui and Tang dynasties in the 7th and 8th century. These became part of the central functioning of the Imperial Medical Academy. They fell out later, but for about 500 years, they were a very important part of imperial medicine. Forms that you see today include Tai Chi, the famous slow-moving shadow boxing martial art, and also individual actions called Qigong, where you will repeat one move over a number of times, and perhaps in a sequence in order to circulate the qi. Okay, so there are many other methods, but we don't have time to go into them today. So in terms of diagnosis, there's 4 main approaches when the patient comes into the office. You'll look at them, then you'll listen or smell them. You get a general sense. How they sound. Do they have strange odors, either that reminiscent of ash or of rotting meat or different flavors, and those can indicate conditions of the state of the body. Then you do patient interview. You ask them about their condition, and these will include a whole range of questions. And then touching refers to both palpation of sore spots on the body, as well as the pulse diagnosis. Pulse diagnosis is a beautiful and fascinating part of the clinical encounter. And you press on these points here on the side of the wrist, on the veins there. And where at these positions that you see indicated, the uppermost points that are closest to the ball of the thumb, just below the crease of the wrist, correspond to the upper part of the torso. So the lungs and the heart. And the middle position corresponds to the middle, so the liver and the gallbladder and the spleen and stomach, whereas the lowest position corresponds basically to the kidney. The kidneys have an important function. And the pulse really on both sides of that, both wrists, reflects the state of the kidneys. So these are kind of the four classical diagnostic methods. Now, when we talk about the disease theory that comes out of this, I talked about the relationship to the seasons, and we have 6 primary external pathogens: wind, heat, dampness, dryness, cold, and fire. And these are particularly effective on certain organs, depending on their correspondence to the 5-agent system. How am I doing for time, by the way? What time should I be? How many more minutes do I have?
Yeah, I think you can go for 5 to 7 minutes.
Okay,
Wow.
Great, good, perfect. Okay, so then the internal pathogens are these emotions that I described earlier, which correspond to the different organs. So joy corresponding to the heart, mind, anger to the liver, anxiety to the lung, worry to the spleen. And so it's after assessing the state of the body, then the practitioner will consider which of these pathogens are having an effect on the patient's system. And then the practitioner will then go through pattern differentiation. And these are questions about one straightforward one, using the principles of yin and yang, which I started out with. We'll ask, okay, is the disease a disease of excess or of deficiency? Is it one of cold? Is it one of heat? Is it internal? Is it external? Is it yin? Is it yang? And these are kind of the Bajet, these 8 principles you organize and take into account all of the different signs and symptoms that the patient presents with and boil them down into a single diagnosis. The other considerations will be whether it's a disease particularly of the qi or the blood, and we'll also consider about where this disease is located in relationship to which of the internal organs. And some diseases may be specifically related to a channel problem. For example, a hernia, which we may consider today to be the splitting of the abdominal wall and the coming through the intestines. But in fact, because hernias often occur along the the small intestine channel, what they will do is give you acupuncture all along the inside of your leg where the channel travels, in order to cause the lining of the abdomen to come back together and to seal up. So you can use acupuncture to treat channels from quite distant places. Okay, now, so these are all quite ancient ideas. How do we apply them today? And how is it that such a medicine has survived? It's a very rich and well adopted and well integrated in Chinese hospitals. It suffered quite some attack in the early 20th century, but with the rise of Mao Zedong and during the communist era, it became the national medicine. But that story, I should point out, is only the story of mainland China. Of course, in Taiwan, Korea, Hong Kong, Japan, Singapore, Chinese medicine has been, and in Vietnam and Thailand and elsewhere, Chinese, or maybe we should at this point say East Asian medicine, has been flourishing and consistently and still very well adopted. It's very well funded, particularly in mainland China, as you can see in this hospital here, which is from a newspaper article from March, showing doctors processing herbs in order to respond to the COVID epidemic. And I want to share with you this volume here, which is just published, and it's free online open access volume, Chinese Medicine and COVID-19, which was published by colleagues of mine. They will be publishing a short article in our society's journal, Asian Medicine, summarizing the findings of this book. But this book translates some of the major interventions and protocols and trials that have been adopted in hospitals in China over the last 3-4 months. And I started out by saying there's an issue of translation, and one of the things that they describe in the introduction is this problem of how do we talk about these traditional principles in a biomedical context. The patient comes in with iron deficiency. This is a quote directly from the intro, by the way, and complains of iron deficiency, and we use traditional diagnostics to say, well, she has spleen and kidney vacuity or emptiness. How do we evaluate treatment? Do we evaluate based on the tongue image, the pulse image, and the symptoms of cold and fatigue that we recognize as Chinese practitioners? And she says there's 3 main responses to this question. One group argues that we treat yang deficiency until it is resolved according to Chinese medical criteria. Others would, using an integrated medical example, would then use Chinese medical systems to treat, but then look for Western medical signs to confirm. The third group just simply adopts Chinese medical treatments without any consideration for the early concepts of Chinese medicine. And they kind of ignore the epistemology of that. And this is this is an ongoing conversation, and it's one that has produced a kind of rich and interesting, I feel, conversation about how we can better integrate different forms of empirical knowledge. And science has been heavily demarcated in certain ways, and I think there is room for better discussion where we begin to talk across these different epistemologies and understand how people who speak different scientific languages can come into a greater conversation. So I'm going to conclude by showing you one example of this. Here's an appendix. In their appendix, this is a list of all of the formulas that have been used in Chinese hospitals to respond to COVID, in the protocols that they describe, that is. And I've underlined one, which go on to the slide. So here's the end of the list. But then I then went and looked it up on this website, which I now want to send to you all in the chat. So this is slightly interactive part of the talk. Okay, you should see that link now. So I'm just going to go through if you follow the link that's in the chat, you'll see what I'm talking about in my slides. So in this website, which has been developed in Shanghai University in Berlin, they have analyzed the contents of the recipe, and they've also described all of the metabolic pathways that the components within those herbs address. If you scroll down further on the link that I sent you, you'll see they also describe all of the different individual herbs, and you can go to those individual herbs and look up their chemical components. And here we have the ingredients and targets associations based on experiment. And this page that you're seeing is only one of 235 chemical ingredients that are in the total formula. Therefore, the related diseases that might be addressed by these chemistry totals 8,603 different potential treatments that this could address. Of course, not every chemical is in every herb, and one herb already is extremely complex components. So how can we begin to use these in an integrated way? This is the question that I think we stand at today in terms of how the conversations that are now ongoing between contemporary modes of knowing and the traditions, which are still live and useful today.
Okay.
Oops.
Yes.
All right. So that concludes my presentation.
Okay, thank you very much. Manjil. Thank you.
Dr. Baker, we are flooded with questions.
Manjil, Manjil, just a moment, okay. Hello.
Yeah.
Yeah. I have a small request to Dr. Kashun. Hello, Kashun, are you here? Hello.
Yes, Kishore, I'm here.
Okay, Kashun, will you just give us 5 more minutes so that we can have 5 minutes question session to Michael?
Yeah, sure, yes.
Okay, thank you, Kashun.
Okay, Dr. Baker, we first have a question from Jayanti Mishra. What is the difference between medical acupuncture and Chinese acupuncture?
Well, if I understand correctly how the term medical acupuncture is being used, it's what Chinese medical practitioners call dry acupuncture, which is the use of acupuncture needles to stimulate either acupuncture points or trigger points without any regard to the Chinese physiology or epistemology. It's entirely a biomedical treatment.
Okay. The next question is from Diksha Dangi. She says that are diseases influenced by factors like emotions and seasons?
Yes, absolutely. In Chinese medicine, for sure. It's the simple answer.
Okay. Another question is from Gori Das. How the Chinese herb Campharia galanca is used as a medicinal herb to cure diseases? Campharia galanca.
That's a great question. I don't know off the top of my head, but I welcome you to drop me an email. I'm going to drop a link in here, MPNS. I'm just going to the acronym in the chat box here, but you can find out what kind of texts cited as what kind of medical systems use the herb by going to search on the Kew Gardens Medicinal Plant Name Services. Just look for Q MPNS, and you can find out more about it there. But feel free to drop me an email. I'll look on my database for Chinese uses.
So participants, we are having a time constraint, so we'll provide you with the mail of Dr. Baker. You can ask your questions there. So Dr. Baker, the organizers of this colloquium would like to thank you for your enlightening contribution on Chinese medicine. He gave us a holistic view of the ancient system of medicine, introduced us to the concept of chai, and elaborated how this system of medicine developed over the years. It was really an enriching experience for all of us. We were mesmerized, Dr. Baker. Thank you so much.
You're very welcome. Thank you so much for the opportunity.
Okay.
Over to Joy, sir.
Thank you, Dr. Gupta. Now, we would proceed to our next speaker, if he is ready. Dr.
Yes.
Thapa?
Yeah. Yes. Okay.
I'd request Dr. Thapa to take over and introduce him, our next speaker.
Okay. Yeah. Kushun, are you there? Okay. So I have a great pleasure in personally introducing our next speaker, Dr. Mudita Kashun Migakumbara. He is currently the head of the Genetics and Plant Breeding Division, Coconut Research Institute, Sri Lanka, and he's a PhD in Natural Sciences from Kunming Institute of Botany, which is under the University of Chinese Academy of Sciences, and he has about more than 17 years of research experience and in the field of plant breeding and molecular biology. He has published an array of papers in reputed journals. He is also a recipient of distinguished Toastmaker Awards from the Toastmaster International USA and also the Presidential Award, National Research Council, Sri Lanka. I now invite Dr. Kashun to deliver his talk. Dr. Kashun, please.
Thank you, Kishore. I'm trying to share my presentation, but then
Yeah, we could see.
you can see my presentation. Can you?
Yeah, just now it has gone out. Otherwise we could see just before this. Yeah.
Can you see it now?
Right.
But then
Yes, sir, you are perfectly visible. Thank you, sir.
it cannot be seen in the window.
Is it so? Hello? Papa? Is that okay? Yeah, we are seeing it properly. It's quite visible.
You can see it?
Yeah.
Okay, shall I proceed then?
Okay.
Right.
Yes, sir, please.
Yes. Thank you very much, Kishore, for inviting me for this wonderful colloquium. And today, I'm going to talk about the tea story, the origin, domestication, dissemination, and utilization of tea plant in Asia. My presentation is started with introduction and then materials, methods, Then I'll be going to talk about the domestication of tea plant in India and China, assessment of tea germplasm in Asia, conclusion, publications, and acknowledgements will be followed at the end. So as my previous colleague, Dr. Baker was speaking about China. Again, I did my PhD in China. It's a diverse country rich with cultural diversity as well as biodiversity. And I did it in Chinese Academy of Sciences. The main university is in Beijing, and my research station was in Kunming, Yunnan province. And I had a wonderful team to work with. I had a colleague from Kenya, Dr. Moses, and another colleague from China, Li Miao Miao. Together with them, we did a global project on tea germplasm evaluation, conservation, and then utilization activities. We did a global project. And my supervisors, Professor Li the Jew was my main supervisor, Professor Lian Mingao, Professor Jian Chushu, and Michael Moller from Royal Botanic Garden, Edinburgh. They were my supervisors. With this background, let me come to the actual presentation. As you know, tea is the most popular beverage, non-alcoholic beverage in the world. China, India, Kenya, and Sri Lanka are the leading producers, and about 85% of the global tea production comes from Asia. And it has discovered with many health benefits, such as prevention of cancer, HIV, heart diseases. Not only that, even for COVID-19, they are using tea as a medicine. So economic value, important cash crop for China, it's about $5 billion. For Sri Lanka, it's about 1.5 billion. And for Kenya, it's about 1.35 billion. And if I tell you a bit about the classification, it belongs to the family Theaceae, genus Camellia, section Thea, species, single species Camellia sinensis with 3 varieties, Camellia sinensis, or the small leaf tea, which is known as China type. And then the big leaf tea, Camellia sinensis variety Assamica. It's big leaf tea basically coming from India. And then Camellia sinensis subspecies Lasiocalipes. It's the intermediate type between small leaf and then big leaf tea. Due to high heterogeneous nature, it's difficult to use morphological, biochemical, and physiological markers for characterization. Hence, molecular markers are the way forward. Domestication is a process that started about 13,000 years ago that brought us to this very colloquium, because that was the foundation which gave us food, shelter, clothing, civilization, and wars. Human age on planet Earth, or the Anthropocene, made a lot of changes and brought a lot of good things as well as bad things. Of course, if you want to read more, there's a book by Gerald Diamond, Guns, Germs, and Steel, and also there was a Nature magazine series in 2015. And then about the origin of domestication of tea plant, still it's a controversy. China, according to the books, it was the place where tea was first used about 2,737 years before. Firstly as a medicine and then later on as a beverage. In the first monograph of tea by Lu in 780, they say the Sichuan is the original area for tea plant. Stout in 1990 said the mountain range from Yunnan to Assam is the original area for tea. And Kingdom Bird also found out, and he suggested that from a primary center, it moved downwards, which I will come to this point later on in my presentation. And then Mayer in 2012 said that domestication of tea possibly in Southwest China around 3,000 years ago. And then if I tell you a bit about dissemination of tea plant, well, there's famous tea horse road which connected Yunnan to the Tibet and even to Nepal. and possibly India via Tea Horse Road, and then even it moved downwards to Myanmar, and then to Cambodia, to Laos, and many other countries. And then the first physical evidence of drinking tea was recently discovered during the Han Dynasty 2100 years ago, and then transport of tea via Tea Horse Road to Tibet about 1800 years ago. Yet, the total dissemination of tea plant in Asia is not known at the moment. So if we look at the published literature about the genetic diversity of tea plant in Asia, it more or less confined to single countries rather than looking at the Asia as a broad level. Only 2 publications have been made in the earlier, yet they have their own weaknesses. And then breeding history of tea plant in Asia is not understood. So today, in my presentation and my study, the primary objective is to investigate the genetic diversity and relationship of tea cultivars, land races, and close wild relatives in Asia using 3 marker systems: simple sequence repeat, and then chloroplast DNA markers, and then using the next generation sequencing technique, single nucleotide polymorphisms. So we have used 3 marker systems to do and come out with answers for these questions. And then we are going to investigate the origin of domestication, breeding history, and dissemination route of tea plant in Asia, and to generate SSR and plastic DNA-based database for cultivated tea and its wild relatives in Asia. If I come to the materials and methods, we have collected 654 tea samples, 589 cultivars, 65 wild samples belongs to 13 wild species from 14 countries in Asia. So the sample collection was not that easy because we didn't just collected the samples from tea gardens. We even went to tea forest and we had to climb up trees to get the samples, and different locations across China and many other countries, 13 more countries in Asia, we got samples for this study. And also we got samples from the ancient Tea Horse Road. This is the welcome I got when I visited there. And then we collected samples from this location too. And then we used 23 SSR primers for all 654 samples, and selected samples we used some of the chloroplast markers, and then the next generation sequencing technique known as genotyping by sequencing was used for 113 samples to come up with single nucleotide polymorphism markers. So these are the techniques that we use for genetic diversity, genetic structure, genetic relationships, and genetic relationships using chloroplast DNA sequences, demographic modeling, and tea breeding history analysis. A lot of softwares I have used, but then I will not go into detail because this has to be a general presentation. So if I tell you a bit about the assessment of tea germplasm and domestication history of tea plant in India and China, no study have been done comprehensively compared Chinese and Indian tea germplasm prior to this study. All samples were genotyped with 23 SSRs and selected samples with chloroplast DNA regions to study the genetic relationships and domestication history. These are the sampling locations. We have collected a total of 402 samples. And then this is across China. We have collected small leaf tea, or from here onwards I will call China type tea. And then from this part in Yunnan, we have collected big leaf tea from China. Morphologically, it is similar to the big leaf tea in India. how its genetics looks like, I will tell you in a minute. So we have collected big leaf tea from China in the Yunnan province, and then we have collected big leaf tea from India, from Darjeeling and Assam area in India. So if I move to results and discussions, when we do the SSR genotyping and then analyze the data, interestingly we found small leaf tea, whether we collected it from China or India, they clustered together, showing that they are same genetic entity. That is, small leaf tea was introduced from China to India. However, the big leaf tea, which is originally classified from India, which is known as Assam, represent wild group. And similarly, we did even the chloroplast DNA sequencing, and again we found same three centers. This is a small leaf tea in China, big leaf tea in India, big leaf tea from China, which shared a lot between the wild types. When we did even the next generation sequencing, again, we found the same evidence. Wild group separates Chinese small leaf tea, Chinese big leaf tea, and Indian big leaf tea. So here in this study, using multiple evidence, we showed that 3 distinct genetic groups in cultivated tea across Asia. This diagram shows that this is the wild species. This is the Chinese small leaf tea, Indian big leaf tea, and Chinese big leaf tea. Very clearly, 3 distinct groups once again, wild separates. So if we look at the dissemination of tea plant across Asia, SSR and chloroplast DNA sequencing analysis shows that Chinese tea domesticated in China and then directly introduced to almost all the tea growing countries in Asia and even in Africa. Chinese big leaf tea or Chinese Assam tea, very limited distribution in Yunnan and neighboring countries, especially Laos, Thailand, Vietnam, Myanmar, and ancient tea horse road possibly was helped in distributing them to downwards. So tea horse road possibly was involved in distributing this tea germplasm. And then Indian big leaf tea, after its discovery in 1823, probably the East India Company or the British, they must have taken the germplasm, which is there in most of the South Asian countries and even to the Indonesia and Malaysia. So if I show you a graphical overview, this is the small leaf tea growing area in China, and from here it has taken to Japan and then to India, to Thailand, Vietnam, many places in Southeast Asia, and even to Pakistan and to the rest of the world. So this is about small leaf tea in China, or sinensis tea in China. Big leaf tea in Yunnan and in China, via tea horse road, it might have gone to Myanmar, Thailand, Laos, and Vietnam. And interestingly, one accession is there in Sri Lanka, and also one is there in Taiwan. If we look at the Indian interesting big leaf tea germplasm from India, it has taken to almost all the countries in the world. It has taken to Sri Lanka, to Southeast Asian countries like Malaysia, Indonesia, and even to Pakistan, to Africa, and many, many other places. We have even evidence to show that it's taken to Africa, because this study has another colleague who look after the tea germplasm in Africa, and then he did a lot of work in African tea germplasm, and he showed that tea was taken from India, Sri Lanka, and China to Africa, and it was distributed from there onwards. So this total study represents about 24 global tea growing countries, and it shows how tea was spread from China basically and from India to the total world of tea growing countries in the globe, and represent about 95% of the tea growing areas we covered during our study. So in conclusion, my study generated the most comprehensive molecular marker data set available to date on tea plant using simple sequence repeat, chloroplast DNA sequencing, and genotyping by sequencing marker systems. And then it's a great asset to tea breeding in Asia. For the first time in this study, we identified that Chinese Assam tea defined as a new genetic lineage. And also for the first time, because there were a lot of controversies in the past, for the first time we have given the due recognition for Indian Assam tea germplasm, or the big leaf tea germplasm in India. We showed that it's independently domesticated in India. And also we showed 3 domestication centers for small leaf tea or China tea, Chinese big leaf tea, and Indian big leaf tea or Indian Assam, independently domesticated. And then we not only found out the domestication events, but then we went back on the time when it was domesticated and how the populations were split in the history of time. And we found that about 22,000 years ago, during the last glacial maximum, species would have moved downwards from their natural distribution habitat, and then in different environments, they have evolved to different genetic lineages. And about 2,770 years ago corresponds well with the earliest use of tea in Yunnan. So this is how the domestication timelines have taken place in the history of time. Not only that, my study found that Cambod type tea, or in the classification it says that Camellia asamica subspecies Laciocalyx, it's a hybrid. And also we found that 2 species, Camellia grandibrachyta and Camellia formosensis, is not agreeing with the current classification system. So there's need to revise the current classification system. And then we also showed the necessity to conserve the Chinese big leaf tea germplasm. And also this study we showed the tea breeding history and role of dissemination route, how it has shaped the current population genetics of tea in Asia and even in African region. So if I tell you a bit about the global significance of my study, here, for the first time, we showed that Chinese Assam tea or Chinese big leaf tea is a distinct genetic lineage, different from Assam tea in India. Interestingly, 55 to 60% of the global tea production, especially the black tea production, comes from Indian Assam tea. So, dear colleagues, if the breeding programs throughout the world, if they can incorporate this diverse Chinese Assam tea into the global tea breeding program, it will increase the hybrid vigor, yield, and quality of the tea plantations across the globe. And these are our publications. We published very recently 5 articles in peer-reviewed high-impact journals, and then 2 articles are in pipeline, and also 2 articles in refereed journals we have published. And this is while I did my thesis defense there, and some other sweet memories in China. And these are the people I would like to acknowledge. Firstly, to Chinese Academy of Sciences, and for my supervisors, Professor Li, Professor Gao Lianming, Chu Jianchu, and Michael Moller. And special thanks goes to Professor Kishore Thapa from India, our colleague, and for all the others who helped with tea sample collection from the World Agroforestry Center. And then another special thanks goes to Dr. Sailesh Rajnikant from Nepal for his help in ecological modeling, and my colleagues Moses and Meow Meow, who helped me a lot to complete this work for my PhD. Thank you all. Thank you very much for inviting me, and if you have any questions, queries, or comments, please, you can ask me now.
Thank you, Kashun, for your interesting talk. Just before giving my official thanks to you, I would like to invite any questions. And I have just received in my chat box that our previous speaker, Michael, with very interest in your topic, would like to ask something. So, Michael, could you please put your mic on mute?
Oh.
Yeah, you can ask.
Hi. Oh, I posted it in the chat box. I had basically 2 questions. One, I think this is really interesting research. I'm interested in the migration of Chinese medical materials. I'm mapping the description of these distributions in textual records, but I can't read phylogenetics. So what advice do you have for someone who cannot cope with the phylogenetic work? Yeah, do you have any guidelines for botanists, someone who's not a botanist or geneticist, to get a sense for the state of the field?
Well, of course, botanical descriptors and everything, like because we work with the Kunming herbarium and all the samples are there, we have deposited them. So of course, yes, we can go there and then get the samples, because even the herbarium specimens are there. But then nowadays, if you have a bit of touch in genetics, then you can do wonders, I'm sure. At least if you can have a colleague. Of course, morphological features sometimes vary with ecological factors. So there can be slight variations in morphological features where genetics come to play, and it can show you the exact picture. That is why people use molecular markers. So I'm sure you can work on this, yes, with the help of a group, maybe, possibly, yes.
Okay, thank you.
Okay, next question I have from Dipayan Banerjee, and he wanted to know the chromosome number of T And one more supplementary question is there any bad effect of tea? We are having tea. Sometimes we have a notion that it will affect our health or something like that. So he wants us to know that.
Well, chromosome number is 16, and we have not found any bad effect of tea.
Yeah.
Basically, because it's used a lot, and even for COVID-19, they said like even in China they use a lot tea, drinking, and then they say that it can help to reduce the infections. Yes.
Right. And there's one more question from Jayanti Mishra. And the question is, why is loose tea is better than the tea bags?
Well, loose tea, of course, then you can see the color and then texture, everything. You can see the quality by your naked eye. And then when it comes to tea bags, of course, they can add chemicals, and then while processing, they can do various things. And they can modify, enhance the qualities, but then which is not there in the real tea. So maybe that is the reason.
Okay, one more question that's regarding your research findings. Is what type of application or future scope that we can have in India, China, and Sri Lanka?
Well, yes, so very good question, because here in this study, we found that Indian Assam tea is different to the big leaf tea in China. So possibly we can get some material by bilateral agreements and start breeding, because then we can have cultivars with very good qualities. Of course, Indian big leaf tea has great qualities. Possibly we can enhance the qualities and also enhance the yield, because it's totally different genetic material.
Okay. Another one question from Arindam Bhattacharya. It has just come. Is there any role of tea in treating the osteoporosis?
Well, there are publications, number of publications. If you just Google it, then you can see 101 publications of different usage of tea. But then, I'm not that expert to answer this question, because there are publications, there are findings, but then since we have not scientifically validated those publications, so Kishore, sorry, I'm not very good person to answer this. Thank you. Yes.
Okay, these are some of the questions that we have received, and I think we also have had the shortage of time. So on behalf of the organizers of this colloquium, I would like to thank Dr. Kashoon for his engaging lecture and on tea germplasm in Asia. And he lucidly elaborated how the different tea species diverge from a common ancestor and a common region. And well, his lecture is especially interesting for the people of our region, because we live in a tea belt, that is North Bengal, our college and your university, and this area in West Bengal. These are the area right from Darjeeling up to near Cooch, Behar. We have tea belt, and mostly all the students of this region, and as well as the faculty, especially of the plant science and the chemical sciences, and even the animal sciences, they will have, I'm sure, they will have a very interesting lecture of yours. And with these words, I again thank you, Kashoon, and it's my great pleasure and also a kind of nostalgia that I'm meeting Kashoon here after 2013, right? And maybe 2013 we met twice there in China. Okay.
Yes, sure.
Okay. Okay, we had the publications together, and thanks goes to Kashoon for everything and whatever efforts that he has done in China, and also he's carrying a very bright research activities in Sri Lanka. Thank you, Kashoon. Over to Dr. Devashish Das.
Okay. Thank you.
Now we have reached the end of today's webinar. Now, two-day international colloquium began this morning with the opening session as the first part of the day, one with the presence of Honorable Vice Chancellor of Cooch Panchanan Barma University, Dr. Dev Kumar Mukhopadhyay, and the Registrar, Dr. Abdul Kader Safeli. Their presence was as significant to us as they were also the chief patron and the patron of the colloquium. We convey our sincere gratitude to both of you, sir, our teacher in charge, Dr. Amitabh Datta, our distinguished speakers from 3 different countries of the world, Dr. Pravir Kumar Haldar, Dean, Faculty of Council of Science, Cooch Panchanan Barma University. He is one of the members of our advisory committee. And above all, the attendees, especially the teachers and research scholars from this country and the neighboring country like Nepal, Bhutan, and Bangladesh. And also, I have heard that there are some participants from USA as well as Kenya. And our beloved students from various colleges and universities. We all believe that science always responds to the needs and interests of the societies. It cannot be ignored that we are all influenced by the cultures in which we grew up and the societies in which we live. Each culture shapes our expectations, values, beliefs, and goals, and also decide the path of science in many different ways. In today's technical session, the lectures delivered by our honorable speakers covered the origin of life and RNA world by Dr. Shah. The second speaker, Chinese medicine, a 2000-year-old living tradition associated with herbs, acupuncture, and moxibustion. Their applications and functions not limited to China but spread the other East Asian countries like Japan, Taiwan, and Singapore by Dr. Michael Stanley Baker. And the last speaker, Dr. Mudita Kashun Meghakumbura, discussed the science behind our favorite beverage, tea, and explained us nicely about tea germplasms of Asia. For us, this talk is so special because tea is the most important plantation crop of North Bengal and many household livelihood dependent on tea plantation. His lecture really boosts the students and researchers of plant science in our region and across the globe. So all the issues delivered today, I think, are well connected with us and our society. We are sure that all the attendees keenly felt interested in the topics presented by our distinguished speakers. Thank you all for your patience hearing for long time. Tomorrow we will meet again at sharp 11 a.m. Have a good day, stay well and stay safe. Hello, Kishore da. Hello. Thank you. Hello, Kishore da.
Yes. Okay.
Yeah.
Okay. I'm here. Okay. Okay. Thank you. Thank you, Ashish. Okay.
Is there any other program actually?
No. Yeah. It's over now. It's over now, and I think we are exactly on time. On behalf of the organizing committee, I thank all the persons here today. That means starting right from all the honored speakers, Kashun, Mike, my two friends, Kashun and Michael are here. We met several times in China, and even in Michael, we met there in Korea. And then one more gentleman, a young scientist from USA, Rana. He's also like our student only. And then to all my organizers and all the participants, especially from overseas participants and also all other participants. We have the cross section of participants, not only from the science background, also from language and literature, and also from the social sciences. All of you, you have made a valued contribution by asking some of the questions to our honored speakers, and also you have actively listened and actively took part in all the lectures and also the presentations and all. And my thanks goes to Mr. Joy Mukherjee and all others who are present here, and as well as all the big personalities from our universities. Yeah, on behalf of the organizing committee and also on behalf of our teacher in charge, who is the chairman of our organizing committee. Due to his illness, he is not able to attend today. Maybe tomorrow he will be attending. And on behalf of all, I declare this international colloquium closed, or I call to the day for today. And tomorrow right at 11, we'll be meeting again. Thank you all. Thank you.